are very specific to vaccinium species, which is cranberries and blueberries. And when you look at the symptoms, this is the symptoms of, this is the cluster of two different varieties. This is Draper here, and this is the blossom blight, which is come early in the season, where you see some kind of blighted clusters, and followed by the infection. You can see that the fruit become prematurely coloring, and they also drop as soon as the pathogen get colonized. So this is the aurora. This is another symptoms we are seeing in the close-up. You can see all these young fruits are really infected and falling down. So it's probably causing a lot of yield losses on the tree, and also later it can really spread towards the downwards. So infection comes on the top of the flushes, and the pathogen travels downwards and infecting the whole stem, and the whole stem dies. And also you can see this is very dramatic in some of the varieties, especially Drapers, and the difference between Phomopsis canker and also the Goudonia canker, which is another canker-causing pathogen. The symptoms is different because for Phomopsis, all the leaves drop off, so you don't really see the attached leaves. But the Goudonia, you can see flagging kind of symptoms. All the dead leaves, brown leaves are still attached. So that's kind of it's not really a clear cut, but that's probably one of the reasons you can differentiate those two. So this is again the infection going all the way down here. You can see the crown region also finally get infected. Once the infection travels down, then all the new growth coming from new stems are also, you can able to isolate the pathogen. So it shows that the whole plant is really, so replanting probably the only way to really address these questions, because fungicides won't help at this point. So this is another plant that's dying all the way down. And this is one of the heavily infested non-bearing field, because most of the symptoms sometimes you see is the non-bearing fields, because the fungicides never, you don't apply any protection for any of the diseases. So they are exposed to these pathogens coming in, so they are not protected. So young fields, mostly the young fields are more susceptible than the mature field because of the production practices. When you cut these stems open, you can really see the browning of the some of the vascular systems, and also some of the pith areas also getting brown. So it's one of the reasons you can really identify this, the pathogen is really going internally colonizing. And another diagnostic tool is to, if you scrape the bark of the stems, you can see the discoloration of the in the internal tissues with green tissue as well as the brown tissue. So it's showing that the vascular system is getting infected. So these are the structures are calling the pycnidias. When you have at the peak of the season, these pycnidia mature and produce spores, and these spores are dispersed by the rain events, usually by the rain events. So these are the structures we are kind of concentrating and seeing throughout the season. We are going to follow and see what these structures are meant for the disease epidemiology. So if you just the kind of a diagram here, the pycnidia is kind of an enclosed body, and you cut across, you can see the spores inside, and this is we call the ostiole, that's opening of the structure, then spores are released through this area. So this is the one happening in the nature. So the objective is to understand the Phomopsis activity in relation to the season. What is the best time or peak time that pathogen is very active in the field? The number two is to what is the active period of this pycnidia produced and the spores are released during this time, and also what are the conditions that induces this pycnidia formation and the spore release. And the other objective is to what is the critical time of infection of blueberry plants, right? It can, because when we seeing the Pycnidia on the plant and what these spores are when they are releasing and what are the critical time of infection, and also what are the temperature requirements for the spores to germinate. So one of the factors we are concentrating is, okay, the spores are released, okay, they are just going and landing on the plant, and sometimes the spores germinate quickly depending on the species or genus, and they can able to infect within 2 hours, 3 hours. Some take longer time. So if you need longer time, you need to have a leaf wetness or moisture sitting on the plant for a longer time for them to germinate and penetrate. So that would probably gives an idea how we can really manage this disease. So just to we started with some laboratory studies to do what is the active period of this pathogen. This is the classical pathology work we do at different temperatures. I will give the Fahrenheit. I can't relate my brain to Fahrenheit, only I work on Celsius. So it's from 4 degrees to 30 degrees centigrade. That is a normal time from spring to fall temperature. And we just put 6 isolates from blueberries and 2 for the comparative basis. We took 2 isolates from cranberries and studied their growth rate over the period of time. So that growth rate is measured every 24 hours after putting on a fungal-based medium, which is PDA, put it in dextrose agar, and measure the growth rate with time. So we plot the graphs with different isolates. If you see that in Fahrenheit from 4 to 36 degrees centigrade we measured, and just to give an idea in Fahrenheit, these are the temperatures. These are mostly active from 5460 to 82 degree Fahrenheit. So the maximum growth is between that time because they are not active below 8 to 10 degrees. So the pathogen is actively growing, but it is not related to infection. If once the infection is taking place, then the disease progress mostly taking place in the summertime. So that's the maximum growth for the pathogen. They're colonizing and they're growing fast at the higher temperatures. So in general, the maximum activity is between 10 and 30 degrees centigrade, and the optimum growth is between 16 and 28. And there is a difference between the blueberry isolate, which are in blues, and also the red one, the broken lines, the cranberry isolates. They are red. There is a difference between them. The growth rate, they are slower growing than the blueberry isolates. So when you look at the culture in a plate, this is just a typical Phomopsis growing on a PDA plate. And when you see after if you incubate for some time, you can produce these tiny black bodies. These are the Pycnidia, which is already shown on a plant tissue. And if you further incubate, they produce this kind of spores, which is oozing through these pycnidial bodies. These are the ones, we call it conidia. These are the ones that's going to spread and infect the blueberry plant. So to understand what are the best temperature, what are the kind of a critical temperature these pycnidias are produced on the plant. But even though this is done in the laboratory, which is not exactly mimic what is done growing on the field conditions, but we pick the temperatures based on the preliminary experiments. We pick from 12 degrees centigrade to 20 degrees centigrade, which is a kind of a time frame, temperature frame that they are able to produce Pycnidia. And we grow in different conditions. One is 24-hour darkness all the way to 20 degrees centigrade. But in 16 degrees, we found there's a maximum Pycnidia production. And then we look at the time frame of 16 degrees, we adjust looking at the climatic conditions, it almost about 10-hour dark period followed by 14-hour light period. So we try to mimic that condition in the laboratory and able to quantify the number of spores Pycnidia produced during that time. So we have taken 2 measurements after growing for 15 days and 30 days. The results we present here after 30 days of incubation. So if you look at this graph, this is the red ones are the cranberry isolates, the blue ones are the blueberry isolates. So cranberry isolates always produce more Pycnidia than the blueberry isolates, but most of the production is around 16 degrees, regardless of different conditions at 16. The spore production is maximum here, it's around 16 degrees, but more than 24-hour darkness exposure, we can see the 14-hour light period and at least 14-hour near UV light produce more conidia. So Pycnidia. So it means that around that time, the most active production of Pycnidia is happening around that time frame. So this is around going Fahrenheit. This is about 60-degree Fahrenheit. The most of the activity of the pathogen, the production is going on around that temperature. So this is the number of conidia on the y-axis. So it's about 500 at the maximum, to minimum about less than 100 Pycnidia per plate. So this is capture. And since we know that the Pycnidia are produced during that kind of a critical time slot, the experiments were done in 2 years, 2014 and 2015. So this is kind of the whole capture of the phenology of the plant, the different developmental stages during the production cycle. When you start with the one plants go out of dormancy in somewhere February, because 2050 is an early season. In dormancy, then you go through different stages of flowering to fruit set to ripening and dormancy. And this is the maximum temperature, day temperature, and the minimum weekly day temperature, and the rainfall incidence. So these are two peaks of rainfall in the early in the season and the late in the season. We have many rainfall events, and the dry season is very long. Dry season we found in 2015. What I'm saying is that the rain events are very important for the carrying the spores, more than the amount of rain, I would say. So during this time, we found that our idea is to study the pycnidial presence on the plant. We take samples we selected 2 fields for 2014 and 2015, and taken 10 to 20 infected twigs, and then brought them to the laboratory and did some analysis on those twigs, looking at the Pycnidia present on those infected materials, taken about 10 to 20 Pycnidia from each sample per field, and then quantified the amount of Pycnidia present on those which have contained conidia. So even though we have Pycnidia presence on the tissue, doesn't necessarily mean they have contains conidia. So one way to quantify them, or each Pycnidia, whether they have conidia inside the pycnidium or not. So that would give a percentage of Pycnidia having conidia. The second, what we did was we have taken each Pycnidium and then using a tissue lyser and just individually placed in each vial, and then break those Pycnidium and release the conidia into the vial, and quantify the conidia, number of conidia in each Pycnidium. So that would give the density of conidium throughout the growing season, and they can really come up with what is the maximum or the most critical time the most Pycnidia are produced. So each sample, then what we did was correlated that with the different developmental stages of the blueberry plants. And also we have taken the cross section of each Pycnidium at different time of the year. So these samples are taken every week. So we are taken from early February to end of December, beginning of December. So we have a series of samples to go through and take sections across and look at the histology of Pycnidia and study their different developmental stages. So when you look at the graph here, this is the number of percentage of Pycnidia on the y-axis that contains conidia. And this is the sampling time starting from early April to early December. These are the different stages of the phenolic plant developmental stages. When you look at the Pycnidia, with the percentage of Pycnidia having conidia, there's not much difference. Most of the Pycnidia we examined, they contain conidia. However, towards the end of the season, we can really see there is a slight decrease, about 50%, only about 50% of the Pycnidia contained these spores, conidia. But when you look at the number of Pycnidia in the conidia in each Pycnidium, the highest number of conidia we found in the very early in the season, it is about 10 to the power 3, that is about 4,000 to 8,000 Pycnidia at the very early in the season. Then during as the season progressed into summer and late in the season, the number of Pycnidia reduced dramatically. Then you don't really see about only few Pycnidia in conidia was found in each Pycnidia. So that indicates that most of the infection is taking place during bud break and flowering time. Then the summer, you don't really see very many conidia. They are almost flat. So it indicates the critical time of infection is taking place very early in the season. So this is the histological studies. We just taken cross sections of all these tissue and look at the pycnidial structures, what it's happening to the pycnidia throughout the growing season. So we can group these pycnidia into 5 different stages. We call it pre-conidial stage. This is the pycnidium you would see when you sample at the bud set and the dormancy time. These are the structures we are seeing, because we don't really see any active Pycnidia with conidia presence. So that indicates the pathogen is producing this kind of structures towards the end of the season as a dormant structures. They remain dormant and then become active early in the season. So this is another structure of pre-conidial stage. This is all found at the end of the season. And then as we see these ones in the early season, we take samples during the springtime, they started to differentiate because these are different names. These are the fungal structures. These are the mycelial parts. They go and produce Pycnidia. The inside you are seeing with the darker cells, they are just beginning to form conidia inside the Pycnidium. So we call it as early conidial stage. And there are 2 pictures of that. And as the Pycnidium mature, they bulk up. They bulk up and push the plant tissue up. And then you can really see a small opening standing up. This is the conidia. Most of the conidia are formed. They're all mature conidia here. And then as we further the season progress, the conidia poses a small neck here, the ostular opening that's about to burst and pushes all the conidia upwards. So these are the conditions we are really seeing during the early season, that is from bud break to flowering. So this is very indicative that most of the infection taking place during that time. So there's another picture of conidial release stage. And then as we go into the summer, these are the ones we are seeing, because these are almost like empty Pycnidium. All the spores are really dispersed. Then these structures, even though we see Pycnidium, that's why we see Pycnidium throughout the year, but those are kind of empty. So that means the infection is not really happening during that time. So these are almost virtually empty Pycnidium there, even though we just see them on the plant tissue. And so if you look at in deeply, so this is the graph I produced earlier with the Pycnidium number, the conidial number in each Pycnidium throughout the season. So during the early in the season, you see these Pycnidia release stages, and the middle of the season, the summer, these are all empty. Then towards the end of the season at bud set and dormancy, these fungus produce, they probably, that's what we see, pathogen and host plants, they co-evolved, they recognize each other. The plant go into dormancy, the fungus go into dormancy too. They produce this kind of Pycnidium, which is really kind of dormant. There's no conidia there. They're ready. Then we go into the next year. They all become active and producing spores. So the other question we had was, even though the spores are released during this critical time of the year, how long that spores takes to germinate. So this is just a simple laboratory test we did to look, taken one representative isolate and took the conidia out, and then studied the germination at different temperatures from 8 degrees C to 24 degrees C. You can see that at the lower temperatures, like 46 and 58 Fahrenheit, the green dots, the germination ratio, this is the mean percentage germination on the y-axis. This is the incubation time on the x-axis, from 0 hours to almost 104 hours duration. So the maximum, we thought that's about 50% germination would give a good indication of what are the best, how long the spores take time to germinate and infect the plant. Most of the, around 16 degrees, as I said, 60 and above, the germination rate is really high. So it peaked about 42 to 56 hours, but 72 hours, most of the, at temperatures above 60 degrees Fahrenheit, they reached 50% germination above. So it indicates, again, even though spores are dispersed, it takes a longer time to germinate and infect the plant material. So that indicates you have a leaf wetness or moisture on the plant is critical. So one of the ways to, if you can really, the moisture is the key factor for germination and infection. So that's another factor we will look into and see whether we can really, what the thing is that if there's a lot of rain frequently in this particular area, then moisture is constantly present. So germination won't be a problem because of the presence of moisture there. So in the summary, I can say the pathogen is very active at 10 to 30 degrees centigrade, which is about 50 to 86 Fahrenheit. The optimum production of Pycnidia by this pathogen was around 16, that is 60 Fahrenheit, with the 10-hour dark and 14-hour light period. The Pycnidia are present on infected plant tissues throughout the growing season, but doesn't mean that they have spores, but they are actively sporulating and releasing spores. The infection takes place only early in the growing season, from bud break to flowering. And we can group this Pycnidia into 3 different stages: pre-conidial, early conidial, conidial and conidial release stages, and also post-conidial stage, which coincided with the different developmental stages of the blueberry plant. And germination of conidia requires long hours, which is equal or greater than 72 hours at temperatures over 60 Fahrenheit under moist conditions. So the information is published in the Canadian Journal of Plant Pathology. So if anybody wants to take a look at it, please visit. So this is just a cartoon just put together for management. This is, the March to this time period is fluctuating because depending on the weather condition, climate of this particular year. So we start with bud break, bud swell to all the way to dormancy. The pathogen is behaving this way. You can see if you start with the dormancy, you have this latent or dormant Pycnidia presence on the plant tissues. Early in the season, they produce conidia. They release during bud break to bloom. Then in this particular area, they are dry. There's no infection going on. But this is the best time the disease is developing. All the blighting you are going to see during summertime because the pathogen is fast growing and colonizing the plant. So management wise, we know that this is the critical time is from bud break to flowering, but fungicide applications are going there. But before that, pruning is really recommended because I have shown those Pycnidia presence on infected tissues. If you prune and reduce the amount of these structures there, then likelihood of having high inoculum early in the season is very much reduced. So it is one of the ways. Pruning is essential for this particular disease. Then you follow up by fungicide applications. But I put some mummyberry, botrytis, anthracnose. All these sprays are going on in the early in the season. So it's not very much you have to just target Phomopsis, but if you have the management tools and time the fungicide application to cover one of these, because mummyberry always starting early, so that means the Phomopsis is also infection probably during that time. So if you space it nicely, then you probably able to cover more than one disease at the same sprays. So this is, even though I don't have much knowledge about what fungicide registered here, this information I got it from Pacific Northwest Pest Management book. For the first column gives the fungicide risk group. Captain is more safer to use, low risk fungicide. Abound is high risk, which is red in between the other fungicide. But when I look at those profile, these fungicides that are registered for Phomopsis also registered for mummyberries, and some of them are anthracnose. So just to give that, you don't have to spray for each and every disease. If you choose the right fungicide, you can cover a wider spectrum of different pathogens at the same time. I think that's my last slide. And I would like to acknowledge two students who worked on this project, Kiko Nabatani and Brandon Wood. And grower participation and the funding is provided by Growing Forward and Agri Innovation Program. Thank you, and take questions.
See, we got a question from Vancouver. What stage of the pathogen life cycle when it is most susceptible? Oh, sounds like you already answered that.
Yes.
Maybe just re-hit that again.
Yeah, the critical time is, you know, that the spores need rain to dispose. So there's no question, either it's early in the season or late in the season. So summer is nothing happening because there's no spores, there is no, maybe there are a few rainfalls, but it doesn't really indicate there's not enough for infection to occur. But susceptible stage is early in the season with some bud break to flowering. And because the plant is, the new growth is coming up and there's a moisture there, a lot of inner spores. So that's the time, I would say, the critical time. But in the fall time, you don't really see spores again because all the Pycnidia produced during that time stay dormant. They don't really develop into producing spores. So the critical time is very early in the season. Any more questions?
Regarding the pruning, do you have to burn those, the pruning, or I mean, if you chop them up, is it still just lying there on the ground?
I think it's chopping would be okay, but as long as you remove that from the field, right? But I have no knowledge whether they're probably going to be active on a dead tissue. I very much doubt it they would develop further because there's no nutrients going further. So pruning would be okay, but as long as you don't keep it in the field. But the question is whether if you have any debris sitting in the field and whether those Pycnidia would further develop and produce spores from there, I'm not really sure about, but very unlikely that would happen. Okay, thank you.
Thank you. Just some wrapping up announcements. We have this session